Okay, so welcome to this video. In this video what we're going to talk about is Cys-Loop receptors, okay, which are also called Cysteine-loop ligand-gated ion channels. Okay, right, so firstly let me give you an outline as to what we're going to discuss in this video. Okay, so the first thing we're going to begin with is a basic discussion of what Cys-Loop receptors are. Okay, so we'll talk about how another name for them is the Cysteine-loop ligand-gated ion channels, and how they're all ligand-gated ion channels that share certain structural features. Okay, in fact they share a lot of structural features with one another. Okay, then what we will discuss is different examples of Cysteine-loop ligand-gated ion channels. So we'll discuss the four main families of Cysteine-loop ligand-gated ion channels in humans, which are the nicotinic acetylcholine receptors, the GABA A receptors, the glycine receptors, and the 5-HT3 receptors. Okay, then what we will move on to is discussing examples from non-human species. So we'll also mention the glutamate-gated chloride channel, which is the GluCl channel, okay, and we'll mention some examples from prokaryotes, okay, so from bacterial forms of life, which are ilic and glyc channels. Okay, then what we will move on to is discussing the structure of Cysteine-loop ligand-gated ion channels in a lot of detail. Okay, and this is where there is a lot of similarities between the human Cysteine-loop ligand-gated ion channels and the non-human Cysteine-loop ligand-gated ion channels, but there are also differences. Again, the key difference is that the human Cysteine-loop ligand-gated ion channels, so the nicotinic acetylcholine receptors, the GABA A receptors, the glycine receptors, and the 5-HT3 receptors, they all have what's called an intracellular domain, as well as the transmembrane domain and the extracellular domain. Okay, whereas the non-human ones, the GluCl channel and the prokaryotic ones, they don't have the intracellular domain, but they do have the transmembrane domain and the extracellular domain. Okay, so what we'll start off by doing is discussing the transmembrane domain, which is the foundational domain after all, because that's the bit that actually forms the channel, okay, for the ions to move through. Okay, then what we'll move on to discussing is the intracellular domain, which is only present in the human forms. Okay, and then we'll go on to the extracellular domain, okay, which is present in both the human forms and the non-human forms. Okay, and then we'll be in a position to actually discuss the gating mechanism, so how ligand binding to these Cysteine-loop ligand-gated ion channels is actually going to cause them to open. Okay, after that what we will do is we'll go into the different examples of Cysteine-loop ligand-gated ion channels in more detail. Okay, and what we'll specifically stress is that the human forms, the nicotinic acetylcholine receptors, the GABA A receptors, the glycine receptors, and the 5-HT3 receptors, they are not individual receptors. You don't just have one nicotinic acetylcholine receptor, okay? They are entire families of receptors, okay? And we'll dwell on that point. We'll look at the different subunits that you can use to build nicotinic acetylcholine receptors, GABA A receptors, glycine receptors, and 5-HT3 receptors. Okay, also what we'll do is go over certain clinical correlates for each. So for nicotinic acetylcholine receptors, we'll discuss myasthenia gravis. For GABA A receptors, we'll look at benzodiazepines. We'll also look at certain heritable forms of epilepsy, which are related to mutations in the subunits for GABA A receptors. Specifically, we'll look at GEFS+, which is generalized epilepsy with febrile seizures plus. Okay, for glycine receptors we'll look at hyperekplexia, and for 5-HT3 receptors we'll look at the 5-HT3 receptor antagonists, which are used as antiemetics and are particularly powerful in preventing vomiting due to anti-cancer chemotherapy. Okay, then finally what we'll go on to is studying the glutamate-gated chloride channel in more detail, which is found in worms, okay, not in humans. And this has a nice clinical correlate in that the glutamate-gated chloride channel is found in parasitic worms, helminths, which infect humans, and therefore there are drugs which can target this channel and can actually lead to the paralysis of the parasitic worm. Okay, and one of the major examples of parasitic worm infection that is treated with these drugs, for instance, the main example being Ivermectin, okay, is onchocerciasis, so we'll discuss that in a little bit of detail. Okay, right. So the first thing then that I want to talk about is what is a ligand-gated ion channel. I want to make sure everyone understands that. So CysLoop receptors are also called CysLoop ligand-gated ion channels, and throughout this video I will be abbreviating ligand-gated ion channels down to LGIC. Okay, so this stands for ligand, that's the L, okay, then gated, that's the G, and then the IC is for ion channel. So because I don't want us to continually write out ligand-gated ion channel, I will be using that abbreviation a lot. So CysLoop ligand-gated ion channels and CysLoop receptors, they're exactly the same thing. Okay, so what I want to discuss then is what ligand-gated ion channels actually do. Okay, and in particular I want to look at their role in neurotransmission and in determining whether a neuron is going to fire an action potential. Okay, right, so basically all the ligand-gated ion channel is it's a receptor for some ligand, okay, and when that ligand binds to the receptor it causes it to open basically. Okay, so they are an ion channel that is gated by the presence of the ligand basically. Okay, and then when the ligand-gated ion channel opens, it will then allow ions to move through it, and that movement of ions often will end up changing the electrical potential difference across the cell membrane, okay, and therefore will cause change in the cell because it has caused that change in the electrical potential difference across the cell membrane. So the response that the ligand induces in the cell is this electrical response basically. Okay, right, so let's now discuss the role of ligand-gated ion channels in neurotransmission then. So let me just draw a picture of a neuron, and I'll draw a picture of the sort of archetypal neuron. Okay, not all neurons obviously look like this, but the archetypal one looks like this. Okay, so you have dendrites coming off the cell body here, okay, like so, and then you have a single axon coming off the cell body, which I'll draw here. Okay, and here is the axon terminal here, and the other side of the axon there, and then I'll draw a few more dendrites here. So there's another dendrite, and there is a final dendrite. And then we'll have the nucleus right in the centre here. Okay, right. So usually in neurons, the role of the dendrite is to have loads of other neurons' axon terminals synapsing onto them, okay, and receiving input from all these other neurons. But the dendrites don't usually actually fire action potentials. Usually the starting point for an action potential is this initial point of the axon here. Okay, and I'll just highlight this up in a special colour. So this starting point of the axon, which I'm now colouring in red here, this has a special name, okay, and this is called the axon hillock, okay, and the axon hillock is the place where the action potential is first generated, and then it will propagate along the axon to the axon terminus. Okay, so the dendrites do not usually generate action potentials. Instead, what's happening in the dendrites is that you're getting currents coming in to the dendrites because of the opening of ligand-gated ion channels, and we'll discuss that in more detail in a moment. And the currents come and have an effect on the axon hillock and either depolarize it or hyperpolarize it, depending on whether you want to induce the axon hillock to fire an action potential or not. Okay, so let's discuss that in a little bit more detail. So basically, although I haven't shown it in my picture, the dendrites would be covered in little structures that are mushroom-shaped, okay, called dendritic spines. So let me show one of these here. Okay, so we're now magnifying in hugely on a dendrite here. Okay, so this is the membrane of the dendrite, and then coming off the side of the dendrite you've got this little structure called a dendritic spine, okay? And the dendrites will be absolutely covered in these little structures called dendritic spines, and these basically are where axon terminals of other neurons can synapse onto the dendrites. Let me just put in an axon terminal here. Okay, so here's some axon terminal of another neuron now synapsing onto the dendritic spine on this neuron here. Okay, right. Now when this presynaptic neuron here fires an action potential, which I'll just abbreviate down to an AP, what will happen is the action potential will make its way into the axon terminal here and cause the release of neurotransmitter from the axon terminal into the synaptic cleft, and it will then go off and act on receptors on the surface of the dendritic spine here, okay, which is called the postsynaptic membrane. And usually those receptors will be ligand-gated ion channels. So if I draw this in more detail now, here is the presynaptic membrane and here is the postsynaptic membrane, so I'll just color code it in. So this is the presynaptic membrane on our axon terminal in blue here, okay, and here is our postsynaptic membrane on our dendritic spine in red here, okay, and you're going to be releasing neurotransmitter into the synaptic cleft from your axon terminal, and what it's going to do is it's going to act on receptors on the postsynaptic membrane, which will often be ligand-gated ion channels, and I'll draw this in the most basic way here. Okay, so this is representing an ion channel, that's the channel down the middle there, okay, and basically the idea is that the neurotransmitter will bind to the ligand-gated ion channel and cause it to open, and then the ligand-gated ion channel will allow currents to move across the cell membrane. Okay, now usually these currents are going into the cell, so usually it will either be a sodium current going into the cell or a chloride current going into the cell. Okay, so usually ligand-gated ion channels are going to either let in sodium ions, so sodium ions will move from the extracellular fluid into the intracellular fluid, or they will let in chloride anions from the extracellular fluid into the intracellular fluid. And the movement of these ions into the cytoplasm of the cell is going to affect the electrical potential difference across the cell membrane. So usually the electrical potential difference across the cell membrane is around -65 millivolts. Okay? Now, if you allow in sodium ions into the cytoplasm of the cell, that is going to depolarize the electrical potential difference across the cell membrane, okay, because sodium ions have a positive charge, okay, and they're therefore going to raise the electrical potential intracellularly, okay. If you let in chloride anions into the cell, that's going to hyperpolarize the electrical potential difference across the cell membrane. Okay, now the basic principle is then that these dendrites all are going to be covered in these dendritic spines, okay, and all over, therefore, the dendrites you're going to be getting sodium currents coming into the cell, and elsewhere you're going to be getting chloride currents going into the cell. Okay, and I should stress that a certain ligand-gated ion channel will only allow either sodium into the cell or chloride into the cell. Okay, if the ligand-gated ion channel is an excitatory ligand-gated ion channel, it will allow sodium into the cell. If it's inhibitory, it will allow chloride into the cell. Okay, so all over the place on these dendritic spines of the dendrites of this neuron, you will have some excitatory ligand-gated ion channels which will be opening in response to excitatory neurotransmitters and therefore allowing sodium into the cell, okay? And you'll also, on different dendritic spines, have these inhibitory ligand-gated ion channels which will be opening in response to inhibitory neurotransmitters and will be allowing chloride into the cell, okay? And basically these currents are going to be coming in and going to the axon hillock, okay, or at least bits of them will be making their way to the axon hillock, okay, and they're going to be summing together. So you're going to be getting the excitatory currents, which are the sodium ions coming to the axon hillock, which are going to be aiming to depolarize the electrical potential difference at the axon hillock, and therefore promoting the axon hillock, firing an action potential. And you're also going to be getting the chloride currents coming to the axon hillock, and they're going to be trying to hyperpolarize the electrical potential difference across the cell membrane. and therefore they're making it less likely that the axon hillock will fire an action potential. Okay, so they sum together, the sodium ions neutralize the chloride anions, and then it's a question of who wins overall, which of these currents is bigger, and if the sodium current beats the chloride current and does produce a net depolarization of the electrical potential difference across the cell membrane at the axon hillock, then the axon hillock will fire an action potential, providing of course that depolarization goes beyond the threshold potential. Okay, so that's the role that ligand-gated ion channels play in neurotransmission. Okay, you've got these dendritic spines all over the dendrites of the neuron, okay, and these have ligand-gated ion channels which will be opening in response to the neurotransmitter being released onto them by axon terminals. Okay, and some of these ligand-gated ion channels will be excitatory, okay, and they will allow sodium ions into the cell, and others will be inhibitory, and they will allow chloride anions into the cell, okay, and these two different currents, the excitatory current of the sodium ions and the inhibitory current of the chloride anions, will then sum together at the axon hillock, and the axon hillock will decide as to whether it wants to fire an action potential or not. Okay, right, so I think that's a really important concept. So now we understand what ligand-gated ion channels then are. They are these receptors for ligands that couple the arrival of the ligand to the opening of an ion channel, basically. Okay, now what we want to discuss is specifically what is a cis-loop ligand-gated ion channel, because this is all rather good, a ligand-gated ion channel, but we are discussing cis-loop ligand-gated ion channels. Okay, so I now want to discuss what is a cis-loop ligand-gated ion channel, and basically cis-loop ligand-gated ion channels are all ligand-gated ion channels that share certain structural features. Okay, now firstly let me just tell you about other names for cis-loop ligand-gated ion channels that are gradually sort of potentially taking over for cis-loop ligand-gated ion channels as the main name for this class of receptors. Okay, so they are also now being called pentameric ligand-gated ion channels. Okay, and for short, pentameric ligand-gated ion channels is abbreviated down to PLGICs. Okay, so if you see that abbreviation, that's what it means. It just means this family of cis-loop ligand-gated ion channels. Okay, and you will understand in just a moment why another name for this family is the pentameric ligand-gated ion channels. In fact, they're actually built out of 5 proteins, basically, so they are pentamers. Okay. Finally, another name that you might see for the cis-loop ligand-gated ion channels is also to call them the nicotine-like receptors, okay, because one of the most notable examples of a cis-loop ligand-gated ion channel family is the nicotinic acetylcholine receptors. Okay, so the entire family of cis-loop ligand-gated ion channels is sometimes referred to as the nicotine-like receptors because one of their most notable examples is the family of nicotinic acetylcholine receptors. Okay, so be aware that there are all these different names which refer to this same family of ligand-gated ion channels. Okay, so now what I want to do then is give some very basic insight to the structure of these things, okay, and we will be going into the structure of these cis-loop ligand-gated ion channels in a lot more detail later on, but for now I just want to give some basic information about them, okay, and a lot of this is going to be oversimplified, and we'll come back and refine it later on. Okay, right, so the first important concept that I want to get across then is that these cis-loop ligand-gated ion channels, they are not, they are not individual proteins, okay, they are in fact pentamers, okay, so to build a cis-loop ligand-gated ion channel, you have to put 5 separate proteins together and stick them together, and that makes you the full cis-loop ligand-gated ion channel. Okay, so I'm drawing the cell membrane now here. So this is the outer leaflet of the cell membrane facing the extracellular fluid above here. So I'll just label this up as the extracellular fluid. Okay, and this is the inner leaflet of the cell membrane, which will be facing the cytosol down here, or the cytoplasm, whichever word you prefer. Okay, right. Now, sitting in the plasma membrane, what I want to show then is one of these CysLoop ligand-gated ion channels. Okay, so whenever showing a ion channel, it's always a good idea to start by drawing a cylinder, so that's what I'm doing. Okay, like so. So this is our CysLoop ligand-gated ion channel sitting in the plasma membrane here, and there's the plasma membrane continuing on the other side of it. Next, I'll draw a little pore going down the middle, which is the pore through which the ions can move when the channel is in the open state. Okay, and now the key concept that I wanted to get across with this picture is that it's not an individual protein. It is in fact a pentamer, so let me now get that across. So it's made up of 5 separate proteins, which I've now split up here, okay, which all stick together to make the full CysLoop ligand-gated ion channel. Okay, so 1, 2, 3, 4, 5. So the full thing is called the CysLoop ligand-gated ion channel. So clearly it is a pentamer. Okay, now an individual portion, an individual protein in the CysLoop ligand-gated ion channel, and I'll just highlight one of these up. So this one here in purple, this thing that makes up 1/5 of the entire CysLoop ligand-gated ion channel, that's known as a CysLoop ligand-gated ion channel subunit. Okay, so understand the difference between a CysLoop ligand-gated ion channel subunit and the full CysLoop ligand-gated ion channel. Okay, right. So you put together 5 CysLoop ligand-gated ion channel subunits, and that will make you the full pentameric CysLoop ligand-gated ion channel. Okay, now what I want to do then is I want to pull out an individual CysLoop ligand-gated ion channel subunit, so let's say this one in purple here, and I want to have a look at its basic structural features. Okay, and the next picture that I'm about to show you is so simplified that bits of it are just wrong, but it will be helpful as a first stepping stone, and then we'll go on to the more complete picture later on. Okay, so particularly be careful about what I'm about to show you as the extracellular domain. Okay, this is firstly just to get a very simple message across, and we will come back and refine this picture basically. Okay, so the first picture then that I want to show you of a CysLoop ligand-gated ion channel subunit is just to highlight the absolutely key structural features. Okay, right. So now this CysLoop ligand-gated ion channel subunit, this is an individual protein. Okay, so it will have an N-terminus and it will have a C-terminus. Okay, so again, this is the cell membrane here. This is the outer leaflet of the cell membrane here. This is the inner leaflet of the cell membrane here, and I now want to show the CysLoop ligand-gated ion channel subunit sitting in the cell membrane. Okay, so the amino terminus of a CysLoop ligand-gated ion channel subunit is extracellularly. Okay, so here's the amino terminus of this CysLoop ligand-gated ion channel subunit. Okay, and then the first structural feature that I want to highlight in this picture is the CysLoop, okay, the thing after which the entire family is named. Okay, so that's why I want to put this in this picture. But be aware that what I'm now showing you as the extracellular domain is so oversimplified that it's just wrong, okay? But there is a point to this picture. What I want to highlight is what a CysLoop is, okay? And then we will come back to the more in-detailed structure of the extracellular domain later on. So there is a lot more in the extracellular domain than just this CysLoop, okay? Right, so I think I'll just stop the picture there then and discuss what a CysLoop actually is first, because since the entire family is actually named after the presence of this CysLoop, it would be quite nice to know what a CysLoop actually is. Okay, so as the little picture here shows, the CysLoop is basically this sort of hairpin loop in the polypeptide structure, which is held together by this bond here, okay? And the reason that this loop is called a CysLoop is that the bond here that holds it together is between 2 cysteine residues, okay? And for short, cysteine is abbreviated down to cis. Okay, so let me show a very blatant picture of this then. Okay, so I'm going to now draw the CysLoop bigger basically. So here comes the beginning of the CysLoop here, like so. So this portion in blue, and this is representing this portion. And then now we're about to see one of the cysteine residues that is actually involved in holding the cis loop together. Okay, so now I'm going to show you a cysteine residue sitting in here. So here is the amino group of the cysteine residue. Okay, like so. So I'm about to show one of the amino acids actually drawn out in its molecular structure. So I've gone from representing the polymer of amino acids just as a line to now I'm going to actually represent the cysteine residue by its molecular structure. Okay, so there's its amino group. Okay, here's the alpha carbon with a hydrogen coming off it. Here's the carboxylic acid group. Okay, and then you'll continue on down here with the polypeptide being represented as just a line again. And now I'll put in the R group of the cysteine residue. So the R group of the cysteine residue has a methylene group, a carbon with 2 hydrogens coming off it, and then importantly it has a thiol group. Now a thiol group usually consists of a sulfur atom bound to a hydrogen atom. Okay, so this is what is meant by a thiol group. And if you're not familiar with the chemistry of a thiol group, I would urge you just to think of it as very similar to an alcohol group, okay, because sulfur is in the same group of the periodic table as oxygen. Okay, it's the atom below oxygen in the periodic table. So it therefore has very similar chemical properties to oxygen, okay? And if that was an oxygen there, we would just call this an alcohol group. So the thiol group has, to an extent, similar chemical properties to the alcohol group. Okay, right. Now, in a pure cysteine residue, of course, then the sulfur should have a hydrogen on here completing the thiol group. However, this thiol group is going to be involved in binding to the cysteine on the other side of the cis loop. So I'm being fortuitous and not putting the hydrogen on there. Okay, then what will happen is the polypeptide will loop round like so. Okay. And you'll have a string of amino acids in between the 2 cysteines here, okay, and in mammalian cis-loop-ligangated ion channels. Now this doesn't go, I don't think, for the non-mammalian ones, but certainly for the mammalian ones, okay, so for all the nicotinic acetylcholine receptors, all the GABA A receptors, all the 5-HT3 receptors, and all the glycine receptors, and I acknowledge that we haven't actually been through all of these types of receptors yet, but just accept at the moment that they are receptors in humans. Okay, and which are cis-loop ligangated ion channels. Okay, now in all of these mammalian ones, the number of amino acids that you have in between the 2 cysteine residues is actually 13, so I might even try and show this. So here's number 1, here's number 2, here's number 3, here's number 4, here's number 5, and they're gradually going to get bigger and bigger. Here's number 6, here's number 7, 8, 9, 10, 11, 12, 13. Okay, so there are the 13 amino acids, and now what we'll have is our next cysteine here. So here is its amino group coming like here. Okay, then we'll have our alpha carbon here. Okay, we'll have another methylene group here, with then the thiol group coming off the side, and then this carbon will also have a hydrogen coming off it. And then here's the carboxylic acid group, which will then be bound to the amino group of the next amino acid along. And now key to this entire structure, these 2 sulfurs of these 2 cysteine residues are going to be linked there. Okay, and this is the key bond that holds the cis loop together. Okay, this is a disulfide bond between these 2 cysteine residues thiol groups. Okay, so this is called a disulfide bond. Okay, so that is what is meant by a cis-loop, and as I say, in all of the mammalian cis-loop receptors, you have 13 amino acids in between the 2 cysteines which hold the cis-loop together. Okay, and we will be seeing what function the cis-loop performs later on, okay, but for now know that this is a feature that all cis-loop ligangated ion channels have in their extracellular domain. Okay, and it's the feature after which all of the cis-loop ligangated ion channels are obviously named. Okay, and in all of the mammalian ones there are 13 amino acids between the 2 cysteine residues. I don't think that holds true in the non-mammalian ones. Okay, I think the number changes, but the principle is still the same, that you've got this loop held together by 2 cysteine residues. Okay, right, so there's our CysLoop. Okay, so that's one of these important features in the extracellular domain. Now that's the only feature that I'm going to show in this very basic picture as being in the extracellular domain. Now that's the part which I'm going to say is oversimplified to the point of being wrong. Okay, we will come back and discuss the fact that there is actually a huge, great beta sandwich in this extracellular domain consisting of 10 beta sheets. Okay, but for now we'll simplify this down and just show this CysLoop, okay, which is obviously a very key feature because it's the feature after which the entire family is named. Okay, right, so then after the CysLoop, what you're going to start having is the transmembrane domains. So here we have our first membrane spanning alpha helix. Now I haven't shown it as an alpha helix because it makes the picture look ugly, okay, but in reality it would be an alpha helix there. Okay, then you have another membrane spanning alpha helix here, then another one, then you have this large loop between M3 and M4, the third membrane spanning alpha helix and the fourth membrane spanning alpha helix. And this is the portion that's only going to be present in the mammalian CysLoop ligangated ion channels. Okay, this is not present in the non-mammalian ones, okay, and that's why they don't have this large intracellular domain. Okay, so this domain that I'm drawing here, this is what's known as the ICL for short, okay, and this stands for intracellular, that's the IC, okay, and then intracellular loop, that's the L. Okay, right, so the intracellular loop is between the third and fourth membrane spanning alpha helices, and it's there and big in the mammalian ones, but it's not big in the CysLoop ligangated ion channel subunits of the non-mammalian CysLoop ligangated ion channels, so the glucial channel of worms and ilic and glic of prokaryotes. Okay, right, so some other key features that I just want to highlight in this picture are the 4 membrane spanning alpha helices here, and I want to give those their names. Okay, so the first membrane spanning alpha helix is called M1. Okay, so this is M1, the second one is M2, the third one is M3, and the fourth one is M4. Okay, just standing for membrane spanning alpha helix. Okay, so these are the M's here. And for this reason, the intracellular loop is sometimes also called the M3-M4 loop because it's in between M3 and M4. Okay, right, so that's the membrane spanning topology then of these subunits of the CysLoop ligand-gated ion channels. Okay, so that's a very basic picture and we will come back and refine that later on. Okay, right. So now what we've done then is we have seen the basic structural features of all CysLoop ligangated ion channels. They are all pentameric like this, and their subunits all have this basic structure. Okay, what we will do in the next video is we'll move on to look at the different examples of CysLoop ligangated ion channels. Okay, so we'll talk about the 4 major families in humans, okay, and then we'll give these examples from non-human species. So we'll look at the glucial channel in worms, and then the prokaryote CysLoop ligangated ion channels that have been heavily studied, which are ilic and glic.